Hello, good afternoon. Uh, Good afternoon, everybody. And this is I'm very excited to interact with young staff members. And I was just waiting. I'm grateful to directors and as well as Dr. Ahmed. And my topic is conventional breeding approaches for meeting the future demands in rice. So we will start now. And by the adoption of the semi-dwarf rice varieties, coupled with better management, there was spectacular advance in rice production during the last 5 decades. So enabling us to a period of self-sufficiency and surplus in rice. And the incorporation of a series of resistant genes conferring resistance or tolerance to biotic and abiotic stresses into the breeding populations, the yield level has been stabilized. So if you'll see the yield gap in rice here, in the research station yield is 100%, and the enterprising farmers, they lose 30%. This is yield gap 1, and that may be due to the environmental constraints and risk-free input management. But the actual realizable yield at field level, we lose about 70%. That's yield gap 2. And that is primarily due to suitable variety, time of planting, time of sowing, modern practices, drainage, weeds, pests and diseases. And through our effort, we can increase the productivity by reducing the actual realizable yield 70% by giving a suitable variety, ensuring proper variety input management, and modern rice management practices. If you see the growth rates of rice in terms of area, production, and yield in Asia as well as in India, that during 70s, we got 0.87 growth rate in area, and the production 1.88, and yield level was 1.01. But by the successful introduction of the high yielding varieties and its widespread cultivation, we saw in 80s, 80 to 90s, there was a spectacular growth in production as well as in productivity. And as we have seen, the area declined, gradually the area declined. And that is obvious that the prime rice lands has been utilized for other purposes, for infrastructure, for housing, etc. But if you see the period from 2000 to 2020, here very abysmal growth rate is there. And this might be due to the changes in the cropping pattern, insignificant increase in irrigation potentialities, and total crop area, and a decline in the use of improved variety and inputs and other inputs in agricultural ecosystem. And if you'll see this one, that there is a declining trend in area and production and as well as productivity. Then if you'll come to the target, that in 2011, our total production was 104 million tons of milled rice, and the productivity level was more than 2 tons per hectare. And in 2020, it was 160 million tons and 2.85 tons per hectare. And the targeted yield by 2030 has been calculated as 145 million tons, and our productivity at the kg per hectare will be 3.45 tons per hectare. So therefore, during these years, during this whole decade for 10 years, that at least we shall have to produce about not less than 3 to 3.5 million tons in order to reach this our production target in 2030. And there is but there are different situations here. Loss of genetic diversity. There were 4 waves of genetic erosion in rice. Our in earlier period, that 82 research stations were spread over 14 districts, developed about 445 varieties by pure line selections by improving the native varieties and from the highly heterogeneous native farmers varieties. And here mention may be made up of GB24, which is derived from Konamani of Tamil Nadu. And this GB24, that pure line varieties, has been very extensively used in the rice breeding program, not only in that area, but also in the national breeding program for its excellent grain quality and as well as higher level of field tolerance to pest and diseases. Then the second wave came, the wide adoption of the semi-dwarf high-yielding varieties, again leading to sharp reduction of genetic diversity. By the introduction of the IR8 and Taichung native one, in the national breeding programs, all were tempted to have our own varieties by making use of the same dwarfing gene, DGWG from IR8 or Taichung native one. And the third genetic loss of genetic diversity, that the hybrid approach, and we have used more than 90% of the hybrids developed, and they use the same sterility inducing wild abortive cytoplasm. And the fourth wave, there was the increased emphasis on the molecular marker assisted backcrossing of the mega varieties that may break down genetic diversity as the fourth wave. Therefore, the loss of genetic diversity is very important factor here in breeding rice varieties. And the important rice improvement programs in India, as well as at the International Rice Research Institute, have not made any significant increase in the genetic yield potential of the varieties released after IR8. And if you fail to stabilize the yield growth level, at least at where we stand today, it may not be possible to sustain the self-sufficiency and surplus in rice. And plant breeders today argue that there is no evidence of yield plateau for any crop, including rice. And it is stated that a perfect variety has yet not been evolved. Still, there is an unexploited genetic variability for the improving the direct and indirect components of yield through utilization of untapped germplasm, and there is still scope to develop and use more efficient breeding and selection techniques for yield improvement in rice. So we'll just start with mating system. That initially the biparental crosses are very widely used in the rice varietal improvement programs, which serve the purpose of combining a simply inherited traits from 2 parents. But here, by the use of the biparental crosses, the recombination possible is restricted, and to make rapid improvement in selfing species in rice, but the use of multiple crosses was suggested by Harlan, Matini and Stevens in 1940, involving 16 to 32 parents, which are crossed in successive generations until the final hybrid involved all the parental genes, 60 to 32 parents. Theoretically, the multiple crosses provide an opportunity for recombination of genes from many parental strains and by intermitting the F1s in successive generations. Now you will find that if you choose here uh, 8 parents, and to get in the final hybrid with all the parental counts, we shall have to double crosses, then quadruple crosses, and finally you will get, and it is in this mating system, it is practically not possible to get enough F1 seed in latter generations of the mating cycle to retain all the parental genes in the final hybrid. That it is assumed that if you take 16 parents and each parent is having a favorable gene, only one favorable gene, so the number of hybrid seeds in each generation ensuring a 50-50 probability of retaining all the 16 genes in one plant in the final hybrid generations would be 8, 64, 131,000 and 64,000 to the power 4 in the first, second, third, and the final generation respectively. So therefore, you can assume here if you involve the 16 parents in a multiple crossing program, the requirement of the F1 seeds to get all the parental combinations in the final generation. Therefore, the second practical limitations was also suggested by Mackay. And he told by using 16 to 32 parents in a multiple crossing program will force to inclusion of a number of unadapted strains which are likely to disrupt the best agronomic property of a variety which has been developed by the breeders and which took several years, even decades, to assemble. Therefore, he suggested modified backcrossing program to obtain optimum number of parental lines, and the unadaptable types should be crossed and backcrossed to the an adapted variety to its before its use in the practical crossing program. So the another way was suggested about the 3-way crosses were tried in many selfing species. The third parent is another adaptable germplasm, and the purpose of increasing the proportion of the adaptive germplasm and simultaneously widen the scope for combining desirable genes from another good parent. And it is observed that the breeding populations of the 3-way crosses had large genetic variance and produce more superior recombinants than the biparental crosses or the 2-way crosses. And such crosses also provide an element of gamete selection, which was given by the gamete selection in maize by Stadler. And he argued since the superior performance of the test cross F1 plants reflects the combination of the superior gametes from the segregating single cross with genotypes of the non-segregating variety as the third parent. As it is argued by Stadler that the frequency of the superior gamete is much higher than the frequency of the superior zygote. Therefore, the 3-way crosses provide a more efficient mating system for obtaining lines with better performance. And besides 3-way crosses, double crosses were tried involving 4 different parents, extending the range of the parents and achieve recombinations from the desirable alleles from the diverse parents. The different mating system as suggested, it may be multiple crosses involving large number of parents, or it may be modified backcrossing with limited number of parental strains, but the major limitations of such mating system was limited to a great deal by the requirement of large number of F1 seeds. F1 generation serves itself as the segregating populations, which is similar to the F2 single crosses, and set the limit of possible recombination of genes available in the populations. And nevertheless, we from these multiple crosses are expected to create an arrow of genotypic variability which have never existed in a breeding populations earlier. And many rice breeders had not used the multiple crosses program because they got adequate success through careful manipulation of the biparental crosses. However, the limited success achieved through various mating schemes employed in rice may be ascribed to number one, the requirement of large number of F1 seeds, relatively small sample size in F2 where the full range of the yield is not realized. And the third point was a majority of the breeders have restricted their selections to known material and have intense effort for a local adaptation. As a result, the certain gene blocks rapidly get fixed along with correlated response, which in many cases go in the negative direction. Therefore, it is suggested to choose the parents on the basis of wide genetic base with acceptable level of productivity and high general combining ability. And before they are used in the crossing programs for realization of high and stable yield in rice. Then I talked here about the multiple crossing in the mating system, about the multiple crossing system in the modified back crossing, and the importance of the three-way crosses, and the importance of
नेट का कुछ प्रॉब्लम है क्या जिमिर साहब?
Yes, I am also unable to hear. से बात करना.
नेट गया लगता है, पूरा बंद हो गया.
He is back. Sir, we are unable to hear you, sir. Hello? We are unable to hear you, sir. So there's an internet issue, sir. He is getting. There's an internet issue, sir.
Is he trying to rectify it?
Yeah, that's it. He's just trying. He's getting it.
Okay. Okay. Good. It can be. Also, it can be their problem.
Yes.
Hello.
Yes, sir. Yes, sir.
Now able.
Yes, sir. Yes. Please, sir. From the gene pool.
Okay. Okay, then I'm sharing
Yeah, please.
this slide.
Please, sir. Please. Sir, kindly put the thing on the presentation mode, sir.
So, so the interspecific oat matings between Male-sterile and the cultivated oat varieties, and Frey and his group over a period of 20 years, they got a significant amount of yield improvement of 3 to 29% in oat. And similarly, an exciting improvement also occurred in Hodium spontaneum, that in case of barley, into the cultivated barley also increased the productivity to the extent of 29 to 46%, and good level of agronomic traits were realized in backcross 3 or backcross 5 generations. And such interspecific mating take the advantages of the nuclear genes from Avena sterilis and as well as Hodium spontaneum and in oat and barley, and had quantum increase in yield potential of the cultivated oat and barley varieties in United States. And the expected proportion of the germplasm switch from the wild species is 6.25% or even less. And in case of both oat and barley, can tolerate this level of wild germplasm for expression of improved productivity. Both the cultivated species and how we can exploit this one in rice. Both the cultivated species of Oryza sativa and Oryza glaberrima and their related species such as Oryza nivara and Oryza rufipogon, Oryza breviculata and Oryza longistaminata have the same AA genome and they share a common gene pool. These weedy species, they can freely cross with the cultivated types. Their F1 hybrids have normal chromosome pairing, but they show some varying degree of levels of sterility. And more often we find these wild relatives has been used in rice for transferring resistant genes into the cultivated types, but never been systematically exploited for increasing higher productivity in rice. Therefore, it is suggested to make use of this weed and wild relatives from the primary gene pool and examine the feasibility of improving the yield potential just in rice, just like in oat and barley, what we have got. And the disruptive mating, that when in near future the plant breeders will be using this wild and weedy resources, and but the whole era of interspecific process are using the exotic germplasm due to the repulsion phase of linkage, which do not express a portion of the potential genetic variations of a trait. And to overcome these genetic limitations, it has been suggested to use the disruptive mating in cereals. And over the cycles of disruptive mating, it is seen that the population mean although remain unchanged, but the variance and the range of frequency distribution increased manifold. And theoretically, disruptive mating by a mating on likes leads to greater opportunity for creating heterozygosity in a selfing generations, because we know by in a selfing species only in the F1 there is a heterozygosity, and that the heterozygosity reduces to have in subsequent selfing generations. And here, therefore, by mating of unlikes enhance rapid crossing over and releases latent variability which is locked up by the repulsion phases of linkage. So the disruptive mating is considered to be the effective plant breeding procedure, as more exotic germplasm will be used in the future plant breeding programs for improving the direct or indirect components of yield. Now in this disruptive matings, from the plus and minus frequency, minus portion of the frequency distributions, they are selected, they are intermated, and if you see it in the different cycles of generations of disruptive mating, that although the mean remain unchanged, but the frequency of the genetic variability variations is wider enough, and releases latent variations which is locked up by the repulsion phase of linkage. Then comes the male-sterile facilitated composites, another method of increasing genetic variation in the composite method of breeding, which was suggested by Sunesan in 56, that he composited all the F2 of different crosses and grown them, subjected, the whole population was subjected to natural selection in diverse environments. An excellent effort that he could, exposing through bulk populations of breeding, that he could able to get the Harland varieties, which was the 28th generation seed lot of this Sunesan's experiment. So, but the major defect was only the combinations where genetic recombination was confined to parents, two parents only. Biparental F2 crosses were composited. So therefore, he introduced a genetic male sterility populations into these breeding populations to become higher yielding in later generations. And several composites developed, which has been verified by Jane and Sunesans, which increased the genetic variability, was not accompanied by the corresponding increase in productivity. The proportion of good recombinants from undesirable linkages increase up to F4 and F5 through additional recombinations and can be isolated in later generations. Therefore, in the earlier generations, the selection was not effective, and there was a recovery of better recombinants in the latter generations. And this is an evidence that Sunesan could get the isolate the Harland varieties, which was the 28th generation's seed lot of his composite cross bulk. So the higher recombinations, it helps by breaking of the parental linkage groups, may help to form a better base populations for the development of the best yielding and higher yielding, highly homozygous and homogeneous lines. And different methods has been used using genetic male sterility. Here is ICAR-CIARI scheme has been given by Singh and Ikas in 1981. And here you can find here that we can go for normal development through pedigree method, and simultaneously we can use these male sterile proportions and introgress different donors of pest and disease resistance to the breeding populations, and the outcross F3 will be grown. There will be selections, and we will get the, from the composite populations also we will derive varieties with better segregants. And the recurrent selections, the initial idea of recurrent selections was independently given by Hayes and Garber in 1919, and East and Jones in 1920. But the term recurrent selection was first coined by Hall in 1945. It's a usual program for 100 years in corn, in wheat also in China for more than 30 years, in rice, the mainstream of breeding methods for upland rice in Brazil for 20 years, and also recurrent selections has been used in barley, Canada. And recurrent selection is defined as reselections generation after generations with the intermating of the selected plants to produce the population for the next cycle of recurrent selections. And here what happened? It increases the frequency of the favorable genes for quantitatively inherited traits of the population of the plants. It is cyclic, and in each cycle there is two phases of plant breeding. That is, number one, selection of a group of genotypes that possess favorable genes and mating among the selected genotypes to obtain genetic recombinations. Therefore, by such intermating, and we will selection the gene as well as the genotypic frequency has been increased. But the major obstacle to genetic recombinations among the alleles at closely linked loci in self-fertilizing species could be eliminated by the use of male sterility factor. And there are many procedures has been utilized by using genetic male sterility factor into the breeding populations, as a result of which will enhance creation of the heterozygosity and create a genetic variability by genetic recombination at the repulsion phase of linkage, breaking the linkages. And similarly, the recurrent selections populations using the male sterility systems and by manual crossing has been also used by the Chinese scientists. And here this is very ICAR-36 male sterility has been used, and gradually, particularly where we find there is no source of resistance, and that is resistance to stem borer. And here by these recurrent selection schemes, continuously we are making recombination at a close linked loci. So the whole populations was grown under pest pressure at maturity and selected resistant plants and select for elevated level of resistance in case for resistance to stem border. And male-sterile facilitated recurrent selections projects has been used by different international institutes like CIAT, Warda, IRI, for tropical upland japonica, lowland tropical indica, upland and irrigated tropical high altitude and irrigated temperate japonicas. And different composite populations were built up through male-sterile facilitated recurrent selections, and these composite populations like CAN, IRAT 4 and 5, and CIRA-CA, CNPF, etc., and in Indica japonica and also Indica japonica gene pool, and which were distributed worldwide to the interested breeders to have their local selection strategy and development of the varieties under different environments. But the major drawback here, by the use of the male sterility factor, gradually in breeding populations in different segregating generations and as R1F2, R2F1, R3F1, gradually we find there is decreasing sterility component. And when the sterility component declines, then there will be the chances of genetic recombination is restricted, because we are collecting the open pollinated seeds from the male sterile plants. And this is the major disadvantage of the male sterility. The genetic male sterility has a selection disadvantage in the populations, and its frequencies decreases to a very low value within few generations. So the effectiveness of this mechanism in promoting heterozygosity and with increased crossing over is soon lost or ephemeral. And there is also some evidences that too much crossing over is detrimental and for the fitness in a selfing species. So another system has been used, that diallele selective mating system or a DSM system. And this system I will quickly cover. The conventional bulk population breeding for the biparental diallele crosses, mass selections in each population series, recombination of the selected genotypes, and introgression of the new germplasm into the breeding populations at any time. So Jensen suggested that the use of male sterility factor to facilitate outcrossing and growing of the breeding population in a specialized environment to maximize genotypic expression of the trait under selection. And this is the scheme has been suggested by Jensen. At any stage now, we will find a line or variety, and we will have a diallele selective series 1 and selective series 2 by intermating of the ones, and finally we will get the breeding populations where better lines can be recovered. So therefore, now we have found this Jensen diallele selectiveness was not experimentally verified due to its cumbersome process involved in different generations and the intermating between the selfing species, even with the advent with the use of the genetic male sterility. Then the next approach comes the history of the stay grain indica germplasm. That the Taiwan japonicas were used as the donors for Hojo Blanca virus resistance, and these parents japonicas have green foliage and little or no leaf senescence at great maturity, and it was repeatedly used, these stay green lines, for 40 years by crossing program, resulting in typical indica germplasm having Hojo Blanca resistance and stray green leaves. And in Colombia, the first say green indica variety was developed as Federos 50. And none of the many varieties produced in Colombia except this variety, only a single variety, and which provides that, indicating the breeders have paid no attention to these stay green traits. And it is rarely observed any stay green plants indicas in other tropical breeding programs also. In rice, all leaves, including the flag leaf of the stay green lines, remain functional past grain maturity, and the leaf color and degree of senescence are inherited independently of maturity period. So one experiment where in a senescing indica variety, a senescence inhibitor gene from Arabidopsis was transferred, and as a result of which it increases due to stay green trait its photosynthetic rates, chlorophyll content, and which gains in tiller number, seed set, plant yield, and biomass. And the stay green Federos 50 at grain maturity showed large advantage in chlorophyll content, photosynthetic rates, and nitrogen content. Now you can see the stay green even after the grain filling period, post flowering and during grain filling period, and at the time of physiological maturity, the plant, stay green plant remain green. And that's what's the advantage. The total biomass it produced is of greater importance. That Federos 50 quickly occupied a large Colombia rice area due to its stay green trait. And it has dark green leaves and unusually long erect flag leaf, non-senescing foliage, and involving an unusually high photosynthetic rate and enhanced chlorophyll and nitrogen content, contribute to maximum yield capacity, extraordinarily high ratoon crop yield ranging from 7 to 10 tons per hectare, superior milling quality, enhanced carbohydrate translocation, and tolerance to stress and stress-related diseases. But here the breeding hello. But here there are many stay green plants, they are not high yielders. So the breeding objectives that the all stay green plants must be combined with superior plant type to give higher yield potential. Selection for stay green trait combined with large and erect flag leaves is a valid and potentially valuable breeding objectives. And at least the basic studies of the inheritance of stay green, its physiology, and its relation to yield potential and other economic traits to increase the efficiency in breeding. Selection of the parents and elite line should be rated for leaf nitrogen maturity, chlorophyll content, and photosynthetic rate before its use for practical crossbreeding purposes. programs. So the next one is the development of the heterogeneous. It is a part of my PhD program that I can tell you here that in case of IR8, when it was released very quickly, it spread over the whole of Asia. And similarly, Gamania, another wheat varieties which was developed in the Western Australia, that also was very famous in the a long time in many areas in Australia. And that is primarily due to some amount of latent heterogeneity which was present in the populations. Here, the cycle here, that it is also possible that this which is not in conflict of the usual pedigree system of breeding, but these apparently uniform lines on the basis of their maturity, grain type and height, etc., they were blended in different populations to have the heterogeneous populations. And it is estimated that these populations, which is there is relatively heterogeneous but morphologically homogeneous, will give a longer varietal life and better yielding ability also. And this is a case studies now that the sub-1 QTL, you all know about the submergence breeding program by the introduction and successful integration of the sub-1 QTL into different mega varieties throughout the world, that BR11 from Bangladesh, Sihrang from Indonesia, Savitri from India, IR64 from IRI, and Samba Masuri from India, Swarna from India, and TDK from Laos. And along with under normal conditions, it is evaluated. It is surprising that except BR11 and its sub-1 and TDK and its sub-1, none of these sub-1 introgressed material were superior to their recurrent parent under normal planting conditions, not under stress. And also, although the sub-1 QTL has been successfully introgressed into different mega varieties, but in terms of survival and regeneration, you can find that they also differ. And therefore, here also the sub-1s, they do not perform better under stagnant flooding situations. And therefore, it is suggested that in order to have a submergence and stagnant flood program, we will integrate that conventional breeding with that of the molecular breeding, first screen for submergence in deep water tanks, and from these desirable crossing programs, and the latter generations we will test it for apply mass for sub-1 and other diseases, and test for yield under normal and stagnant flooding conditions, and as a result of which the convergence of conventional breeding and molecular breeding, we can develop varieties tolerance to submergence as well as tolerance to stagnant flooding. And there are different target traits. I am not explaining this one. And now we come there. This is very interesting slide here, that I have taken the grain number here in one axis and 100 grain weight in the other axis. When the number of grain number was increased, the frequency of the high-yielding genotypes were increasing. But when the grain size was increasing, 100 grain weight, and the frequency of the high-yielding was not there. But interestingly, you will find where there is moderate expression of the grain number, moderate expression of the 100 grain weight, and moderate expression of the panicle length and panicle, that accommodated large number of genotypes here. So there in the breeding program, any breeding program, no, we should not select for extreme types. We will select the plants with fair combination of different traits, as we have seen here. And these genotypes are were expected to give a better yield, simultaneously greater stability of production in the different environments. So this is the a trait, that is number of grains, then the grain weight, strong stem, number of panicles, and etc., which has been routinely carried out at the plant breeders, conventional breeders, to select plants for higher yielding ability. And the convergent breeding program here, there is stepwise improvement. If we'll find to develop a unique variety, there will be stepwise improvement gradually. And besides yield, better resistance, tolerance to biotic and abiotic stresses can be achieved, although it takes time, but it is possible to get it. And about the rapid generation advance, I am not going to explain you everything, but here the speed breeding we talk about here, nearly breeding for photoperiod sensitive rice varieties in the tropic. Generally we take 1 generations per year, but by the rapid generation advance there will be 3 generation year can be grown through RGA. And here there is higher genetic gain by modifying the empirical formula used by the plant breeders. That is, the genetic advance is the selection intensity, heritability coefficients, and phenotypic standard deviations divided by L. L is the length of the breeding cycle or generation interval. Here the genetic gain is increased due to reducing the time of the breeding cycle with the breeding method used SST with coupled with rapid generation advance. And future projections, many methodologies has been used in several decades, and we shall have to re-evaluate the use of alternative breeding methods like bulk population or single seed descent, and which may reduce the labor and resource and speed of the breeding. Fixed lines, early generation selections for yield also considered, and better exploration of especially the PBS trial data can be used, which is underutilized by the breeders. And the use of new technology or breeding informatics, and the breeding informatics method including more efficient experimental design, which is such as row column design or partial replications or spatial analysis, single site trial analysis, etc. And major emphasis will be given to the multi-environment trial, and it will be important to improve classification of the target environments in order to maximize efficiency. Say for example, that is the characterization of the flood water, geographical information systems, and environment classification should be useful. The molecular breeding will continue to improve the efficiency of the breeding program, and profiling of the breeding material with specific gene markers, as we have seen in the sub-1, and a suite of other traits. And this could be, it is hoped that the marker should be used more routinely in early generations. However, the cost is prohibitive due to large number of samples, and the use of molecular breeding may be more effective when multiple abiotic and biotic stress genes or QTL segregate within specific populations, because multiple target loci can be screened in these populations. And the synergy between breeder and molecular genetics. This is very interesting. Rice breeders have been very successful in selecting for a wide range of traits using only conventional methods. And breeders do not need to use markers for every trait. Even the perfect markers are available. The full advantage of mass can be realized when the markers are used to do something that cannot be done using conventional methods, such as marker assisted backcrossing, MABC, or marker assisted field admitting. And what is the knowledge gap and application gap between the conventional breeders and the molecular biologists? Many breeders may not understand the topics in the molecular breeding in sufficient detail. And similarly, the molecular genetics often do not understand the breeding processes to identify the most strategic stage to apply markers. This also prevents the integration between breeding and molecular genetics, including sharing of the germplasm, such as mapping populations or sharing information or trait data. And the application gap Breeders are usually focused on developing new varieties where the molecular genetics are focused more on the identification and discovery of gene and QTL and ultimately publications in high-rated journals. Furthermore, the location of the breeding and molecular genetic research is often separate and may occur at different institutions. Therefore, our future need will be planned and well executed QTL mapping experiment, QTL and marker validation activities, efficient genotyping systems, cheaper genotyping systems, and breeder-friendly analytical tools, and integration between breeders and molecular genetics will be critical to develop new germplasms using markers. These are my reviewer teachers. He is Professor H.K. Mahanti, from whom I learned the art of plant breeding. He is no more. And he is the living legend, Professor E.A. Siddiq. And students were his life. At any moment you reach him, a new emerging problem and its solutions will come up. And this is Dr. McKill. And at ICAR-CIARI, I was working with him. He is the father of the QTL, Submergence sub-QTL. And it is very pleasant to work with him. Very friendly. And the next one, this is a very pleasant moment while getting the Senadera Award. And you can find here the physiologist, Dr. Ismail. And I told several times that the physiology and breeding should not work in isolation. They must be working jointly to have some productive output. And I have learned through over years that in difficult moments behave like a duck, keep calm and unruffled on the surface, but keep paddling away like crazy underneath. And this is harvesting joy, which is a gentle reminder that life is a cycle, that which is sown, it must be reaped. Advice to the next generation of the plant breeder. While there are indeed many difficult challenges ahead in the 21st century, but you shall have to remember that a minor yield increase of 1% will have, in a widely grown variety, will have a tremendous impact. Even in the advent of the new promising DNA markers, such as DNA technologies, such as DNA markers, the time-tested conventional breeding and selection approaches will still account for significant contribution and persist for another 50 years to come. A sound theoretical knowledge coupled with considerable hard work and long hours in the field are the secrets of breeding success. And this is likely to be the case for many years to come. The success and sustenance of the rice breeding tales. There is no easy way to rice improvement. It demands patience, dedication, continuity, and our total mental and physical commitments to field work. So there are two more slides, which is very interesting. It is although not associated with the subject, but I always tell that science without philosophy is not science. So these two memorable incidents here, Sri Umashankar Joshi, former Vice Chancellor of Vishwabharati and President of Satya Academy Jnanapitha Award winner and famous Gujarat writer. He has an opportunity to see Mahatma Gandhi after 21 days after his fasting. And when he saw his eyes, his glowing eyes, that there was the shadows of glimpse of love and affection to the mankind. In 1945, 6th August, atom bomb was dropped near Hiroshima. But before that, I can tell you, when he went to Japan and they saw him a heap of black ash over white dots over that, and they told them that it is our monument. Then he told, are you not criticizing me? Is it your monument? They told, yes, sir. And here, this place, a huge marriage ceremony was attended by thousands of people. And about 1 kilometer away from this place, atom bomb was dropped. The radioactivity instantaneously killed all men and women attending the marriage ceremony. Their flesh bones melted into a heap of black ash. The white dots represent the melted bones. And the newly married couple, out of fear, them they embraced each other and their body melted, and in one corner of the house their body melted and imprinted as a wall as a silhouette art. And this is really the two contrasting features of human behavior. One is the emblem of love and affection, and the violence, heterogeneous, and cruelty to mankind. This is a natural phenomenon which is occurring now. And this becomes a kaleidoscope through which to view the reality in life, the way to relate to our world, and the way we live to work and work to live. And another, during this World War II, another interesting slide is here. This image of 1945 during World War II. A Japanese boy stood in front of a cremation pyre, waited for his turn to cremate his dead little brother, Joe O'Donnell. The American photographer who clicked this picture said the child was biting his lips so hard to prevent crying that blood was dripping down in the corner of his mouth. When the guard asked, "Give me the load you are carrying on your back," the child replied, "It is not a cargo, he is my brother." In Japan, even today, this image is used as a symbol of strength. So painful, yet so inspiring. And nothing is more beautiful than a real smile that has struggled through tears. And the passions in my life, I found intense happiness, I found anguish, I found madness, I found loneliness, I found solitary pain that gnaws my heart. And three passions have governed my life: the longing for love, the search for knowledge, and the unbearable pity for suffering of mankind. This has been my life. I have found it worth living and would gladly live it again if chances are offered to me. Plant breeders, they just a moment. The plant breeders never retires in life, and his stories will remain untold without you all. My best wishes and kind regards. Thank you very much.